Um, so the spread of attention within objects has been proposed as a neural mechanisms of how humans or primates can group features and be able to perceive objects. And it is a mechanism for object-based attention. To simply see that, if the monkey was supposed to fixate that point, the red dot, and find out what is the other dot, what is the other red circle that is attached to it, you would imagine that it would spread its attention in order to group that curve. When you look at the receptive fields in V1 that are closer to the point of fixation versus receptive fields that are farther, you would see that the activity elevation in V1 is sooner for receptive fields that are closer to the point of fixation. So to interpret that, you would say that the activation is a spreading, that in a way, the elevation of activation happens along the path of the object. And that's the mechanism that a lot of people think would help segment objects. But how would neurons be able to impact each other that way? How would they be able to actually impact in a way that could spread the elevation of activation? So when we look at the surface of V1 and the colors show orientation selectivity, and this is superficial layers, people have observed this long-range horizontal connection from orientation columns that are coding for the same orientation. And the suggestion is that these long-range horizontal connection, they actually mediate what we call association fields. Association fields were suggested in that paper, and they look something like that, where neurons that have similar selectivity could form associations, and they could basically modulate each other, up-modulate each other, while they down-modulate the neurons that have slightly different orientation selectivity. These association fields, you could imagine, could really help you segment in a task like this, right? Because the way that the segmentation would happen, the spread of attention would happen, is through collinearity, right? The cells that have collinear response would be able to up-modulate one another, and then the activation can spread through the association field in V1. But what if you're dealing with something like this? What if you're starting from that center dot on the zebra in the front, and then you want to spread attention within that object? You really cannot use association field in V1, because if you actually are following the collinearity, like the type of signal that you have in V1 or the type of association field that you have in V1, you're not going to spread within the object. The attention is going to go along a strip. The question we wanted to ask is that, can we actually use association fields? Can we actually extend this concept beyond V1 in order to be able to spread attention within objects? So Gilbert and Lee suggested in a review paper a while ago that association fields are not just in V1, but this could be something far more ubiquitous in the brain, that you have these associations being made in different stages of the visual hierarchy and also in frontal area and parietal areas between different entities. And we think there is some truth to that. Now, if we want to study associations, right, we have to use the right tools. Right? If we were studying hierarchy, if we were studying a V1 type feature selectivity for Gabors, well, convolution is the right thing to do. If you are studying associations, I think transformers are the right tools to use, because they have this built-in inductive bias where they actually learn associations between entities, right? If they're processing text, you could imagine they're learning associations between the words in the sentence. In this case, well, the word it would be associated with the animal. The coloring on the left shows how strong the association is. And it's through these associations that you'll be able to predict the next word. Well, what would this look like in images? Well, now, what are we associating in the image? What do we want to decide what's related to what? Well, in this case, a simplifying assumption would be to, well, let's divide it into grid, to patches. That way we could see, well, how similar is this patch of the image to another patch of the image? Ideally, at some point, this would move on to proto-objects and superpixels that are kind of informed by the visual properties, but initially this works as well in practice. If you divide the image into patches, then you could say, well, after encoding them, how similar are these patches to one another? And then they could impact one another. They could impact one another across the hierarchy. Now, models that are trained for classification in this manner, as if you've trained model for classification, you know that the representation collapses around the categories, and they're not super useful for feature extraction. But there are other models that are trained with self-supervised objectives. It could be Dino, which is distillation with no labels, or it could be mass autoencoders that you may have seen already in the conference, where you're not trying to classify anything, but the objective is much more similar to the objective that large language model use, which is basically filling in the blanks. When you do that, you'll get representations for each unit. Now, if you get representation for each unit, for each patch of the image, you could actually compare those representations, right? Association fields are trying to associate different parts of the image with one another. So we're thinking that we could model these higher level association fields through the affinity of these representations from different parts of the image. It's very much like association fields on the lower level, right? On the lower level, you're considering orientation features. On the higher level, you're considering much more abstract and rich features. So, as I said, you could divide the image into patches, and then you could look at the affinity signal for a specific patch in the image. Now, if I take that patch that is on the front zebra, I could take a simple cosine similarity, the angle between that vector representation and all the other patches in the image, and then see how similar they are. And that would give me an association field, which basically says, well, how similar is this representation of this patch to the other patches? I could look at another patch. Sometimes, like in this case, because I'm looking at a patch on the zebra's ear, then it would be more similar to the other zebra ears in the image, or another patch. Now, what to note, but is very important, is that these affinities are very dynamic. If the image changes, well, these affinities completely change. If you move your eye or if you move your head, the world changes, and the affinities in these different areas of the image change. So they're very dynamic, not static like how people think of some of the V1 affinities. But the question to ask really is, are they object-centric, right? I'm showing that patch at like on the zebra. What's important for the association in this case to be able to help us spread within the object is that it has to be object-centric. How would we measure that? We could take a random point in the image on the bottom left, look at its association field, and then ask the question of, what if we start with a very high threshold and lower our threshold? And then we could look at the true positive rate and false positive rate. And then as you lower your threshold activity, you could see all the areas that are highly associated with this area and light them up. When you do that, you could see that the true positive rate goes up, which is the activity within the object over the area of the object. And then as you lower your threshold, you could see that the activation outside of the boundaries of the object comes up too. If we get the true positive rate going really high up before the false positive rate increases, then that shows that it's object-centric. We tested the different models. We saw that object-centric representation. We saw in some of these models that if you look at ResNet, for example, you would see that ResNet is really not object-centric, meaning that if I look at the convolutional filter, which is height, width, depth, and I'll take one column in that and then take a correlation, take a cosine similarity of that column with all the other columns and do the same analysis, you'll see that it's not really object-centric, mostly because convolution is a fixed representation. It doesn't have the dynamics of associations that transformers have. You could also look at across the layers of a network and see how object-centric representation, the representation becomes more object-centric across the 12 layers of a VIT, a vision transformer. Now, in order to test our model, I'm going to show you a behavioral study that we collected data from, and it's a simple same different paradigm. You have two dots appearing on the image, and people have to decide whether the dots are on the same object, in these two cases, or a different object. And we made sure that the distances are similar, so there is no signal whether the two thoughts are on the same object or not based on distance. The behavioral result that we saw is consistent with what people have seen in simpler stimuli, where there is the same object advantage. When the dot is on the same object, people are faster to respond, but also the effect of distance only exists in the same condition. Again, the interpretation of this, like I showed you with the simple line, is that the attention is spreading within the object. It reaches the closer dot faster than the farther dot, and then it goes and says different. So that's the process, and that's how you get this behavioral pattern. There's variability for each condition that I'm not able to get into, but it's interesting. So it's not just all one, but rather within each condition, you could have higher or lower reaction time. So we devised a very simple model. We have association fields, so we could just say, okay, from this starting point that also people started, what are all those patches that are really highly associated with this patch? And then we spread to those. Then now that we have a growing segment, we could say, what are all the patches that have high affinity to this patch? Then we could spread from there. And if we continue this process, we could see that only through lateral connectivity, we're able to spread within the object. And I could show it in a more dynamic way. This was surprising to me, that objectness, and I think that's a very important point, that objectness can be embedded within the lateral connectivity in higher visual areas in models that are not trained for classification, right? These models are trained for reconstruction or self-supervised objectives, but objectness can be embedded within their lateral connectivity in higher visual areas, and I think it would be really cool if someone would be looking for these in higher visual areas. I think they would be very informative. One way to get reaction time from our model is to say, well, how many steps did the model take in order to reach each of the dots? Now, naively, we could also spread it just until it reaches even the other dot, and we could just draw a histogram of it. We had 255 trials in each condition, so you see the number of trials, and on the x-axis, the number of steps to the second dot. As you would expect, the histogram gets shifted to the right when you go from same close to same far, and then when you go to different close. And the bars that I'm showing on the most right are the ones that the attention never reached the second dot. So that you would expect that would happen much more commonly, as you could see here. For example, in almost 170 trials, attention never left the object, which is very impressive. If we average these responses, we can see that we get a similar stereotypical pattern of the behavior that we saw earlier. We could also look, I'm out of time, but we could also look at individual trial level correlations and show that these models close the gap to some extent, but there's also still a lot to be explained in the behavior from where these models are to the subject agreement. So I think it's exciting to continue working on this project. With that, I thank you and my collaborators.
Thank you. Please, time for questions.
Thank you for a very interesting talk. One thing that you said is that objectness is implicit in the connections.
Yeah.
It seems to me these visual objects have explicit representation somewhere. If I understand correctly, the way your model works is that there is this transformer processing the image, and then there is some Python code or something inspecting the activations and spreading these things. So do you have ideas about how this might actually be implemented in a more biologically realistic manner? And am I on the right track in interpreting your model?
Exactly, you're perfectly right, yeah. So I think you could imagine, so I'm trying to say that different parts of the image are not just a single cell being represented, but rather imagine a hypercolumn. A hypercolumn is a collection of, let's say, hundreds of neurons. Its activation, it could be described like a vector in a space. So if you look at a hypercolumn in V4, and you have, let's say, 500 cells, you could describe its activation like a vector in a space. So that vector in a space could connect to other vectors in the space nearby based on the angle of the activation between the two. So that's, I think there's not anything implausible about lateral connectivity representing, like there's nothing implausible about cortical columns representing vectors of vectors and also the columns being connected to one another through these sorts of lateral connectivity. I think everything I said is somewhat plausible. The process of getting those activations could be improved, but I don't think it's biologically implausible.
So let's have the next speaker get ready. So in the meantime, and take advantage of my moderator privilege.
Sure.
There was earlier work by Steve Grossberg, who have done a lot of very similar things. Maybe you can articulate what is the difference and what is the novel contribution. He also looked at the V1, V2 hierarchical association, and some of the works mentioned in the V1 salience in my hypothesis in the second speaker also use these isofeature interactions. What are the...
So great work from Grossberg, obviously, but I think what they never managed to do was scaling those models to more complex objects, right? With art model and other models, they're great theoretically, but I think we're kind of continuing on that path, trying to think what does it mean for those models to be applied to scenes, to complex environment that you have looking at this room, right? I don't think they ever really managed to do that, and I think we're kind of making that a step. But theoretically, I think we're inspired by that work.
Thank you.
Yeah.